Hello everyone, welcome to today's live broadcast, Detection of RNAs with Flow Cytometry. I am Judy O'Rourke of LabRoots and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by eBioscience, an Affymetrix business. eBioscience provides innovative, high-quality reagents to researchers around the world, fueling the process of scientific discovery in the areas of cellular immunity, cell biology, and oncology. The company's extensive portfolio of leading-edge cell analysis products and technologies centered on flow cytometry and immunodetection places its customers at the forefront of science. PrimeFlow RNA assay, the first assay capable of simultaneous detection of RNA and protein within millions of cells at single-cell resolution, represents the capabilities and expertise of eBioscience and Affymetrix, brought together to enable the scientific community to advance the understanding and application of biology for a better world. For more information, please visit www.ebioscience.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button lower left. I would now like to introduce today's speaker, Filippos Porichis, the Director of International Programs at the Reagan Institute of MGH, MIT, and Harvard. Filippos serves as a member of the Executive Committee of the MGH Center for Global Health. He is an assistant in immunology at Massachusetts General Hospital and an instructor in medicine at Harvard School of Medicine. Originally trained in biomedical sciences at the University of Portsmouth, UK, he received his PhD from the University of Crete, Greece, and the Institute of Molecular Biology and Biotechnology, and subsequently joined the Reagan Institute for his postdoctoral training under the supervision of Dr. Daniel Kaufman. Dr. Porichis' research at the Reagan Institute focuses on the immunological aspects of T-cell function in HIV and tuberculosis infections and the implementation of novel techniques in order to evaluate the immunoregulatory networks that lead to T-cell dysfunction. During the last 6 years, Filippos has investigated the immunoregulatory networks that govern HIV-specific T-cell dysfunction and have showed that manipulation of inhibitory pathways, such as PD-1 and interleukin-10, can restore function of pathogen-specific T-cell responses. His research showed that HIV-specific CD4 T-cells are regulated by different patterns of inhibitory receptor other than HIV-specific CD8 T-cells. I will now turn it over to Dr. Porichis for his presentation.
Hello everyone, thank you for attending the webinar. My name is Filippos Porichis and I'm a faculty at the Reagan Institute of MGH, MIT, and Harvard in Boston. Today I'm going to talk to you about an assay that we developed in the lab in collaboration with Affymetrix and other groups for high-throughput detection of gene-specific mRNA at the single-cell level with flow cytometry. As I mentioned, I'm a principal investigator of the Reagan Institute, and the main focus of my lab is the characterization of T-cell function in chronic infections. The main idea is to understand the function and the immunoregulatory pathways that regulate T-cell functions in chronic infections. And the main goal is to identify novel targets for immunotherapeutic interventions to improve current therapies or for the rational design of vaccines for HIV and tuberculosis. Now, one of the biggest obstacles for a more comprehensive understanding of how the immune system works in physiologic and pathologic conditions is the availability of technologies that push the barriers of current analytical tools. For these reasons, we develop a lot of effort at the Reagan Institute for the development or implementation of novel technologies that will enable us to fully characterize cell subsets and basically understand how the immune system works. Now, cellular heterogeneity, particularly in primary samples from blood and tissues, is a major obstacle for understanding the true biology of gene expression and function. When we are performing transcriptional signatures or transcriptional analysis with either RNA sequencing or microRNAs or qRT-PCR-based methods, what in bulk populations, what we are measuring is the average expression of RNAs in the population that we have. The reality is that the bulk population that we were looking at, even though we hypothesize it's homogeneous, the reality is that it's not actually homogeneous. So when we're performing transcription analysis, you may have cells that express high amounts of this transcription of this gene, or you may have cells that do not express this gene at all. And what we're measuring with these methods, with RNA sequencing and qRT-PCR, is basically the average of these expression patterns. Single-cell analysis is a rapidly growing field in biomedical research that is expanding our understanding of biological characteristics of human diseases. Now, for immunologists, antibody-based flow cytometry has been the gold standard throughout the years, and it really helped expand our understanding of the functional and phenotypic characteristic of several subsets in physiological pathogenic conditions. Now, advances in molecular biology allowed single-cell transcriptomic analysis, either with RNA sequencing or with methods that are based on qRT-PCR. However, these methods are very laborious. They rely on very specific technical skills, and as I said before, they rely really on sorting cells, either with flow cytometry or with other methods. Now, if we combine all these technologies, we can start having a very good understanding of the biology of different diseases. But the more we develop techniques, as I said before, the better we'll enhance our understanding on how the immune system works in different diseases, and this is basically one of the goals. And if we can identify techniques that can make everyday lab work better, and we can apply these techniques routinely in the lab, this is to our advantage, and we can move our science forward. Now, I will take you through our efforts in developing these techniques. It all started in about 2010. I was still in my first year of postdoc in Daniel Kaufman's lab at the Reagan Institute. And one of our collaborators, Daniel Kavanagh, he was interested in applying this technology called quantigene ViewRNA cell assay for fluorescent in situ hybridization. So it's basically a fluorescent in situ hybridization assay at the time for a company called Panomics. It was still not acquired by Affymetrix. And what this assay basically did is use the FISH, a fluorescent-based in situ hybridization technique, to visualize DNA or localized RNAs in cells that were adhered in cover slips. So you can basically take these cover slips, go on a confocal microscope, and you can visualize your fluorescent labeled RNA or DNA. Now, at the time, the way this assay works is basically you have a first hybridization step where you can have probes, proprietary oligonucleotide probes that bind specifically to the targets of interest. And on average, you can have about 20 oligonucleotide pairs per transcript. Now, then you have a second step where you basically have a signal amplification based on a branched DNA technology. And what you basically have is you have the probes that bind to the gene of interest. Then you hybridize a pre-amplification DNA sequencing, and then the sequence, and then there is another amplification step where you put even another step of branched DNA sequences, and this basically creates like a tree-like structure. And at the end, you can hybridize another set of probes that are now labeled with fluorescent dyes, in reality is Alexa dyes, and you make what I call a Christmas tree, where you can have about 400 labeled probes per transcript. Now, if you combine the 20 oligonucleotide pairs with the 400 labels, you get about an 8,000-fold amplification. So instead of amplifying the transcripts, as we usually do with RNA sequencing or with qRT-PCR, in this system we amplify the signal. Now, as I mentioned, this assay was at the time in 2009, 2010 offered only as a platform for confocal microscopy. And what Daniel Kavanagh wanted to do at the time was he wanted to identify infected cells with RNA transcripts for HIV infected cells. So he wanted to use this platform. And we had recently acquired a confocal microscope at the Reagan Institute, and I was one of the few people at the time that knew how to use it because I was trained in confocal microscopy in Greece. I was by no means an expert, but I was able to use the confocal microscope. So I offered to help Daniel Kavanagh. And at the beginning, we had difficulties with autofluorescence and the samples not being optimal. So we got in touch with a company, and they were always very helpful, Panomics. And as we started talking, I said, listen, I work in a lab with Daniel Kaufman. Our expertise are in immunology. We really do flow cytometry all the time. Daniel is one of the best at the Reagan Institute working on flow cytometry. So our expertise really rely on that. And because the basic of this method that we are using for confocal is on detection of the Alexa fluorescence, the fluorescence of the Alexa dye, why can't we use this for flow cytometry? The reply that we got from the company at the time was that they didn't have expertise in immunology or flow cytometry. They are molecular biologists. They did not have a flow cytometer in-house. They had a confocal microscope, so they didn't have the expertise to really help. Other people have asked the same question, but at the time they were a smaller company, so they didn't have the capacity to help us out. As I mentioned, I was still in my first year of my postdoc, and I was very stubborn at the time. So I decided to embark on establishing this assay just for historical reasons. Daniel Kavanagh was able to get his nice pictures, and they made it to the cover of AIDS Research in Human Retroviruses. You can see here a picture with a confocal picture with HIV infected cells that are basically detected with the HIV RNA. But as I mentioned, I embarked with Daniel Kaufman on trying to establish this assay with flow cytometry, and we developed different methods and different protocols. The main principle is that you have cells that are in suspension. Instead of being adhered on a cover slip, we have cells in suspension. We fix the cells. We fix the cells with PFA. We permeabilize the membrane so the target probes can have access. At the time, the protocol had a protease step, a proteinase K step for better detection of DNA and RNA. And then the rest of the protocol is we have the probes that bind to the DNA. Then we have the hybridization steps, as I mentioned before, with the pre-amplifiers and the amplifiers. And then you make your Christmas tree. It was a very complicated protocol at the time that we tried to develop, and it took a lot of time. But what we were able to do in the summer of 2010, we were able to use this technology for the first time in our laboratory to detect a housekeeping sequence. It's a ribosomal 18S sequence. And you can see here nice detection of ribosomal 18S in PBMCs versus the irrelevant probe, which is, you can see here. You can see nice detection with flow cytometry. So we went back to the company. We said, listen, we can do it. It's doable. We can detect signal. And they got even more excited, so we established a collaboration. So they were providing technical support and some reagents. We were providing our knowledge on immunology and what would immunologists want to detect with this assay. What you can see here, one of the first experiments where we had different probes detecting 18S, for example, with different sensitivities. We now went to other to different housekeeping genes, and we were able to detect that with different probes, probe 6 and probe 8. We perform a series of experiments to optimize the assay with proteinase K dilutions. We identified that 1 in 2000 was the best. I don't want to spend too much time on that. It's just highlight that we spent a lot of effort to really optimize the assay. We optimized different steps for hybridization. In our hands, the 3-hour hybridization was 3-hour time point hybridization step was the best. We also did 4 hours or overnight. Overnight was actually pretty good as well, but we stuck with the 3 hours. So we spent a lot of time optimizing, and in about 2000, in about September, we came up with one of the protocols, one of the first protocols that we employed, and we always wanted to improve on that. And during that time, Affymetrix had acquired Panomics, and the company, they were also working now with other groups in other universities, and really we put a team of experts in trying to develop this assay, because the company now realized the necessity of such assay. Now, even though we work a lot on the assay on our hands, it was very troublesome. The main issue was that when we tried to transfer the protocol that was based on adherent cells, the buffers were very harsh. So we had, they were not optimized for the cell buoyance of cells in suspension, and we had severe cell loss. So at the time, it was not really suitable for studies with rare populations. We would start an assay with 5 million cells, and at the end we were only able to acquire about 2,000 cells. So it was really not optimal. We would get signal and we get nice separation, but it was really not optimal for the studies that we wanted to do, which is investigate rare populations such as HIV-specific T cells or TB-specific T cells. Now, the protocol at the time was very complicated. We had individual probes that we had to mix with the different pre-amplification, amplification, hybridization buffers. Then it had a protease step that was making our life difficult because it wasn't compatible with antibody staining. The signal was not stable, and because the buffers were very harsh, it basically altered the shape and the size of our cells. So we couldn't really have forward and side scatter in the flow cytometer. We could get a general gate, and we could see the signal, but it was very cumbersome. It was promising, but it was very troublesome. Now, later on, we continued working with Affymetrix at the time, and then eBioscience came on board, and they brought even more expertise on fluorescent microscope, on flow cytometry. We continued to work with them. We identifying where the cell loss happens, which buffer is responsible for that, and other groups also, as I mentioned, started to work on it. And a few months later, Affymetrix and eBioscience came up with a protocol. They contacted us and said, "Look, we think we fixed the buffers. We think we have an assay that is now working very nicely, and we want you to try it and tell us what you think." So we got the main assay is basically based on the protocol that we had before. So you have cells in suspension. You stain your cells with antibodies now. You can fix your cells. You can permeabilize them. The fixation is PFA and methanol based with this protocol. And once you've opened holes in the membrane, you can now go with your hybridization probes to detect your RNA. And then you make your Christmas tree with the amplification steps. And finally, you add your labeling probe to complete your Christmas tree, and then you can go on a flow cytometer and you can detect the genes of interest. So as I mentioned, this new kit was based on methanol fixation permeabilization. Now Affymetrix and eBioscience, they develop a novel assay, the PrimeFlow RNA, that is not based on methanol, and it actually allows you for detection of intracellular pathways with antibodies, intracellular markers with antibodies, and I will discuss that a bit in the end. Now, the probes that now the company offers, they have 3 different set of probes, so you can detect up to 3 different transcripts. You have type 1, which is an Alexa 488, an Alexa 647 dye. You have a type 4, which is an Alexa 488, and you also have a type 6, which is an Alexa 750. And you can see here the different sensitivities of the different probes, and you can basically decide for markers that you need sensitivity because of low abundance of mRNA, you should basically try and get the Alexa 647. For transcripts that they have high abundance, so you can easily detect them, we should go with a less sensitive probe. So we got this assay, and we started to, we used that assay for the first time. We wanted to detect beta-2M, and we're really excited that we saw a nice, sharp detection of beta-2M in PBMCs, again in primary cells from HIV-negative subjects. And you can see the difference between the beta-2M and the relevant probe here. So this was really exciting. The next step that we wanted to do, as I mentioned, this assay now, the next step we want to do is, as we've done in the past, is kinetics with hybridization. And we recently had a paper that was accepted in Nature Communication that describes a lot of these experiments and how they were performed. But what you can basically see here, on this side we have an irrelevant probe, DAP-B, that targets a sequence that is not supposed to be present in human samples. And you can see the different time points here. We tried 2 hours, 3 hours, and 1 hour, 2 hours, 3 hours, and 4 hours of hybridization. Now, when we look at the beta-2M, which is the housekeeping gene of interest, what you can see here is that the 4-hour in our hands was optimal, and it gave better results than the 2 hours and the 3 hours, and much better result than the 1 hour. However, for protocol reasons, and we, apart from sensitivity, we also want to look how the protocol is flexible in everyday life, lab work, and we stuck with the 2 hours because it was actually giving us a very nice separation. It was much easier for a protocol to use. So 2 hours in our hands was the optimal time point for hybridization. Now, as I mentioned, this protocol now is, you can use antibodies to stain for different cell types and cell subsets, and this is critical, especially when we're working with PBMCs or samples from tissues or human samples. Now, the Quantigene protocol is methanol-based, so you have to use methanol resistance or methanol-sensitive fluorochromes. You can see here the list. You can also find that on the website of Affymetrix in Bioscience. And unfortunately, this assay was not compatible with dyes, protein-based dyes such as Psi7, PerCP, or APC. It was really good though in our hands with Alexa dyes, with eFluo, brilliant violet, PAC blue, and Q dots work in our hands as well. But you can see the limitations. You can use some antibodies, but you cannot use some other antibodies based fluorochromes. So it has limitation, it has the advantage as well though, compared to other assays. Now the first thing we did is we want to try and see if this assay that in the past, in the beginning, was really harsh with our cells, if it's now compatible with these antibodies, and we know we could use it, but we want to compare it with regular flow. So on the left, you can see staining with PBMCs on the regular flow cytometry protocol with PBS and FCA and with PBS buffers and regular flow cytometry buffers. And on the right, you can see staining using this novel mRNA fish assay that we developed with Affymetrix. And you can see nice forward and side scatter of our PBMCs. We can exclude doublets really nicely. We can use a viability dye. You can distinguish dead cells with live cells. You can gate on CD14 cells, monocytes, or CD3 cells. Here we're gating on lymphocytes, so we don't have a high expression of CD14 cells, so we can gate on our T cells. And then within our T cells, we can now gate on CD8 cells and CD4 T cells very nicely, and it's very comparable to what we see in the same patients with regular flow cytometry. So this was very good news. But at the end of the day, what we wanted to do is we wanted to see if this assay is sensitive enough to detect other genes apart from just housekeeping genes, and if we can now go down to detect RNA expression in HIV-specific cells or CMV-specific cells or antigen-specific cells. So we decided to look at cytokines, because it's something that we were familiar with in the group doing intercellular cytokine staining, with staining either for IL-2 or TNF-alpha or interferon gamma or different cytokines after stimulation with different antigens. So what you can see here, and I'm putting this as you can see it's not going to be the best detection that we had, but I'm putting this figure there for historical reasons because it was the first ever assay that we tried. So what you can see here on the forward scatter versus IL-2 mRNA, stimulation was done for 24 hours, no stimulation, it gave us some background. When we stimulated with PMA onomycin, we were really excited to see that we can detect about 33% of our T cells, of CD4 T cells that can express IL-2 mRNA. We stimulated with CMV lysate. We knew that this patient had responses for CMV, so we were able to detect responses for NCMV. And interestingly enough, we were also able to detect responses for HIV when we stimulated with HIV GAG peptide pools. So this was really exciting. We had IL-2 that we detect here. We also did the same experiments with interferon gamma, and you can see here PMA onomycin, CMV lysates, and HIV GAG peptides in comparison to the no stimulation. So this was really exciting. Now, when we saw this assay first, in our lab meeting, people said, well, how do you know that this is specific? How do you know that what you're detecting is actually specific for the RNA that you're saying you're detecting? And it's just, some other non-specific staining. The advantage of this assay, as I mentioned before, is now you can go and stain for different antibodies. Apart from just staining for example, CD4 and CD8, you can stain for functional markers. And when you stimulate T cells, one of the things that will happen, the stimulated T cells will upregulate CD69, and then you can basically detect CD69 you can detect your the antigen-specific cells based on CD69 expression. So what you can see here is CD69 expression based on the protein versus mRNA expression based on the probe. And for this experiment, we use the Alexa 647 probe, which is the type 4 probes. So what you can see is that we can see nice detection of HIV GAG peptide pools. We can see nice detection of interferon gamma using staphylococcal enterotoxin B as an antigen, and obviously we can see a nice detection of interferon gamma when we stimulate with PMA ionomycin. And it is very important here to state that all the detection that we are detecting, all the mRNA, interferon gamma mRNA that we are detecting basically comes for CD69 positive cells. If this was unspecific, we would basically see detection of interferon gamma from non-CD69 positive cells. It's not a clear story, but this would give us a hint that it's not specific to what we want to detect. But as I mentioned, mRNA did come from CD69 positive cells. Now, we showed that again to a lot of people, and then they said, well, where is your ICS control? Now, as I mentioned, with this version of the assay that we had at the time, we could not do intracellular cytokine staining. So what we were we could not stain with intracellular pathways at that time. So what we did is we took a sample, we split it in two, we stimulated cells with PMA ionomycin. Half of the sample was stained during a regular intracellular cytokine staining, and the other half was stained using the quantitative flow view RNA assay. What you can see here in this first ever assay that we did, again, I'm putting this figure for historical reasons, for when we want to detect proteins, we were able to detect about 60%, and this is overnight stimulation, we're able to detect about 60% of our cells that were expressing IL-2 protein. When we looked at the mRNA transcriptome for IL-2, to our surprise, we saw that only 37% of our cells were expressing the protein at the same time point. And this got us a bit worried. And then you can see another experiment that we have here. You can see IL-2 protein versus interferon gamma protein. And here you can see IL-2 mRNA and interferon gamma mRNA. You can again see that even though we have nice correlation between the populations, we still get lower percentage of what we will get with ICS in this overnight stimulation assay. And then this was a bit puzzling to us. And one of the first things that we started to think is that when we do intracellular cytokine staining, what people will do is you will get your cells, you will stimulate them, and then after half an hour or 2 hours, depending on the protocol, people will add inhibitors for protein secretion, such as either monensing or Brefelding, and then you will keep the cells in the incubator overnight. So what you end up doing is you basically trap the proteins inside the cells overnight, and what you are detecting after when you come the next day is basically the amount of protein that is trapped inside the cells during this time of 16 or 18 hours or 12 hours, depending on your incubation, or 6 hours if you're doing a 6-hour ICS assay. However, with a quantitative flow view RNA assay, we do not add any inhibitors. So you have free cytokine secretion, and then the mRNA is constantly renewed. And so basically what we are detecting is a snapshot of the mRNA at the 18-hour time point, or at the overnight, when we do overnight incubation, at the 16-hour time point of when you fix the cells. So it's not the total amount of RNA that has been produced during the 18 hours or the 16 hours, but it's just a snapshot of when we fix the cells in the morning. So as you can see, there's a discrepancy between an intracellular cytokine staining that detects protein during the 16 hours during the overnight incubation, or with the quantitative that just detects a snapshot. So in order to overcome that, and we really want to prove that what we are detecting is the mRNA that we're detecting correlates with the protein, and as I mentioned before, we cannot, with this version of the assay, we cannot do intracellular staining. So what we thought about doing with a PhD student in a different lab, but collaborating with us, Morgan, what we were able to figure out is this adaptation of the method where we adopted the interferon gamma secretion assay from Miltenai. And how this assay works is that you can stimulate your cells, say for a period of 18 hours or overnight, and then what you can do at the end of this incubation time, you can use this reagent that Milton has developed that basically has an antibody that can bind to leukocytes. I think it binds to CD45, so it can bind to leukocytes in PBMCs. And on the other side of this antibody is conjugated with an antibody that is specific for interferon gamma. And then you can incubate that for 45 minutes, and what will end up doing that the T cells that or the cells that are stimulated and they're secreting their interferon gamma, basically this interferon gamma is then captured in this interferon gamma antibody, so you can really detect now the cells that are secreting the interferon gamma. And then you can go with a secondary antibody for, again, interferon gamma in this instance. That is, the secondary antibody is labeled with PE, so then you can go on the flow cytometer and you can detect the cells that secrete interferon gamma for the last 45 minutes of your incubation. This assay lasts for 45 minutes. Now, using this assay, we're now able to detect interferon gamma protein here and then interferon gamma mRNA using this novel flow FISH assay. And you can see here when we have no stimulation, the background is basically clean. When you stimulate with PMA ionomycin, we can see a nice detection of interferon gamma protein that correlates very nicely with interferon gamma mRNA. It's almost like it's a compensation issue, but in reality it's a very nice correlation of expression. And we found the same with SEP stimulation, and interestingly enough, we found in the same with HIV GAG. So these are HIV GAG specific CD4 T cells in this instance, and we've done the same with CMV specific T cells and also CD4 and CD8 T cells. So this was proof of principle that the mRNA that we are detecting at the snapshot of when we fix the cells actually correlate very nicely with the protein that has been produced with the protein that has been produced during the 45 minutes of incubation with this capture assay. And this really proved that what we are detecting, the mRNA correlate with the protein. So it's very specific. Now, things to consider for immunologists when they want to do staining for cytokines is that, as I mentioned before, ICS, intracellular cytokine staining, is based on detection of proteins that have been trapped inside the cells for a duration of time. When you want to detect mRNA, people need to consider that different cytokines will actually have different kinetics from mRNA expression. So, for example, IL-2 will have different kinetics than interferon gamma. So when you stimulate your T cells, IL-2 will come first depending on the stimuli, and then interferon gamma will come at a different kinetic. So you really need to choose the time point that you're investigating the cell in order to be able to capture the mRNA of interest. Now, again, different stimuli will produce different kinetics of mRNA expression. For example, PMA ionomycin is a very strong stimuli. It will have very fast kinetics. SEB is a less strong stimuli. It will have less kinetics. And then if you want to do peptide stimulation, this has a whole different set of kinetics for mRNA expression. So scientists need to consider what sort of stimulation they're looking at. And even to make things more complicated, different cell subsets will have different kinetics of mRNA expression after stimulation. Naive cells will have a more latent response than effector memory that will have a faster response and will have a quicker transcriptional regulation and transcription expression. So when immunologists try to do these experiments now, they need to adjust the way of thinking, and it's just a 6-hour or 12-hour assay that we do with regular intracellular cytokine staining. Now, as I said before, people said, well, what you're showing now is very nice. It's a novel assay, but you're detecting interferon gamma and IL-2. We can do that with regular flow cytometry. The reality is that, depending on the in vitro system that you want to do, as I said, Berfelding and Monesi will block cytokine secretion, so you cannot actually do a lot of functional assays. T cells, these cytokines actually play a role in communicating and giving signal to different cells. So if one wants to do a functional assay where they need cytokine secretion, they can now, in combination with detecting the cells, they can now use this assay that freely allows cytokine secretion, so you can have the cytokines be secreted and give signals to different T cells or different other cell types, such as monocytes or B cells, and at the same time you can go and detect the T cells that produce these cytokines. But this is not the most powerful advantage of this technology. The most powerful advantage is that this technique is not based on antibodies. You can use antibodies, but you can actually have probe designed for any sequence of interest inside the cell. So we embarked upon proving that, and what we wanted to do, one of the first things, since we're working on CD4 T cell immunology, and we're interested in T follicular helper cells, we had a lot of issues in the lab trying to detect IL-21. So what I'm showing you here is a set of experiments that we did where we used spleen CD4 T cells from subjects that have ITP, idiopathic thrombocytic purpura, thrombocytopenic purpura. It's an autoimmune disease where the body will produce antibodies against platelets, autoantibodies against platelets, and T follicular helper cells have been shown to play an important role in that, and basically by producing large amounts of IL-21 that drives the B cell responses and leads to this autoimmune disease. So what I'm showing you here is a regular intracellular cytokine assay with samples from this patient that we had with this disease. And when you have no stimulation, IL-2 and IL-21 levels are minimal. And when we stimulate with PMA onomycin, we see nice levels of IL-2, but the levels of IL-21 that we detect at this time were very poor. So we decided to use this assay, the new FlowFish assay, to see if we get any improvement. And to our surprise, what you can see here is using this novel assay, we were able to detect 16% of CD4 T cells that producing IL-21 mRNA. This is in contrast to about 1% that were able to detect protein. Now, an antibody did exist for IL-21, as I mentioned before. The way we measure IL-21, though, either the in vitro assay or the antibody is not optimal for that. And I know now there are a new set of buffers and a new set of protocols that can kind of optimize detection and get better better detection than what we got about 3 years ago when we did this assay. But you can clearly see the advantage of this FlowFish assay to detect IL-21 mRNA. And I have a second patient here. We're able to get another subject that had, again, the same disease. And again, we were able to detect nice IL-21 expression with this assay, but we're not able to detect any protein. And just to show how specific this is, IL-21 is supposed to be coming in literature mostly from CD4 T cells. Some CD8 T cells were shown to also produce. But in these patients, when we look at CD4 T cells, we get nice expression of IL-21. When we look at T cells, we get no expression of IL-21, even though we get nice expression of IL-2 or interferon gamma. So detection of IL-21 in this patient and the other patient that I showed you is specific for the CD4 T cells. Now, these patients were ideal. It was one of the first, these samples were ideal because it was one of the first experiments that we did with 3 probes. And this is what I'm showing you here. You can basically use 3 different probes to detect mRNA. And with a very, very nice separation of the population. You can see here it's almost a 2-log separation that we get for IL-2. We get nice separation for interferon gamma. And we get, again, nice combination of IL-2 and IL-21. And here you can see the interferon gamma and IL-21. And all of these are from one sample. So simultaneous detection of 3 different mRNA cytokines using flow RNA. And I think this is very important for the field. Now, moving on, apart from IL-21, we were also working with other cytokines, and in the lab we were interested in IL-10, is an immunoregulatory cytokine that plays important role in chronic infections and cancer and in other fields. And there are several antibodies out there. We in the lab had issues with detection of that, of protein. So we embarked on using this assay. You can see here now gating on monocytes. After we stain with R848 or with interferon gamma and LPS, we get a nice increase in mRNA levels. And at the same time, we got the same sample, we split it in 2, and we performed QRT-PCR. So we again see a nice correlation of detection of IL-10. As I said before, we are interested in immunoregulatory pathways, and IDO is a pathway that has it's implicated again in cancer and other diseases such as HIV and other chronic infections. And IDO is an enzyme that basically catalyzes tryptophan, which is an amino acid that is necessary for T-cell metabolism. So when IDO is up, it catalyzes that, and it basically causes starvation of the environment from this amino acid. And then this amino acid is necessary for T-cell function. So if you have starvation of these components, nutrients from the environment, the T cells basically become dysfunctional. And we were interested in investigating that. At the time when we started the experiments, there was no antibody to detect IDO. Scientists will base their experiments on qRT-PCR detecting mRNA. I know now that eBioscience has an antibody that can detect IDO. I haven't used it myself, so I don't know how it works. But now there is an antibody available. But bear in mind, when we did the experiments, there was no antibody available. So basically what I'm showing you here, and again, this is a figure from the paper that was recently published in Nature Communication, you can see that when we take PBMCs and we stimulate them with different stimuli to stimulate the TLR pathways, you can see that when we get on the monocytes, you have here an irrelevant probe. This is unstimulated cells that have a basic level of expression, and then we get, when we stimulate with R848, we see an upregulation of IDO, which is even more pronounced when we're looking at stimulation within the interferon gamma and LPS. And we repeated that in 5 subjects, and you can see that basically in every subject, every time we stimulate within the interferon gamma and LPS, we get an upregulation. As I mentioned before, there, what we were able to do also is we took these samples and we split them in 2. Half of them were treated with this flow fish assay, the other half were treated with qPCR. So we just got the bulk PBMCs and we performed qRT-PCR. And what you're seeing here now is that a very tight correlation of the level of mRNA that we were able to detect with qRT-PCR in bulk PBMCs with the level of IDO mRNA MFI, the mean fluorescent intensity, on CD14 cells. So now, because we have these antibodies, we can get on the different population, we can really, in PBMCs, and we can really see which population is responsible for this upregulation. And in the paper, we saw that it's not T cells, it was not B cells, it was really monocytes that caused this, it was really in the monocytes that this enhanced expression of IDO mRNA happens, and we were able to correlate that with the mRNA levels measuring with qRT-PCR in bulk PBMCs. Now, coming close to the end, we're wondering what other applications we can have. And as I mentioned, this essay started because of our interest in confocal microscopy. Doing this essay for confocal microscopy, we were immunologists due to the expertise that we had in Daniel Kaufman's lab at the time. So we set up this essay in immunology, but we do understand that being able to visualize your mRNA and proteins is very important. So we wonder if we were able to utilize ImageStream, a technology that is a flow-based assay that basically allows you detection and visualization of your cells and protein detection. You can visualize protein expression. So what I'm showing you here as an in vitro assay is the assay that I showed you before with the capture assay, where we can capture interferon gamma protein along with interferon gamma mRNA. Here we have no stimulation, so basically there is no protein, but you can see down here with the ImageStream, we get nice detection, visualization of CD4 protein, nice visualization of CD3, but there is basically no interferon gamma protein and no interferon gamma mRNA. Now, when we stimulate with SEB in this instance, we now see a nice population of interferon gamma protein and interferon gamma mRNA double positive population. And when we go and look at this population with ImageStream, we can now see CD3 staining, CD4 staining. We can visualize mRNA staining, and at the same time, we can also visualize interferon gamma protein. And this, I think, you know, even though we made kind of a loop, it's again one of the, from imaging going to flow cytometry, with this technology, we can basically combine now both. And if anybody is interested in doing co-localization studies to see if their mRNA is co-localizing with a protein, you're now able to do that. I'm showing you some of the data that we had from this assay that we did before, where basically this is proof of principle. It doesn't really show anything, where CD3 and CD4 staining co-localized, while mRNA and protein at this set of experiments is not localized, not co-localized. As I mentioned, the way we perform this assay, the interferon gamma capture protein is based on the outside, where the mRNA detection is based on the inside of the cell, intercellular. So we do not expect to have any co-localization of this. And this is proven by the analysis that Amy Baxter did in our group. In Daniel's group in Canada. But this is kind of a proof of principle that if someone is interested, and depending on the studies that they can do, they can use this nice technology. Now, coming towards the end, and what we were interested in is look at microRNAs, and microRNAs they have been shown increasingly now to play an important role on T-cell regulation, immune regulation, and different aspects of physiological pathologic conditions. And Affymetrix actually has an assay an assay that can detect microRNAs with the quantigene assay, but it's based on, again, on the confocal assay that they have, on the microscopy assay that they have. And you can see this. You can go on the website and you can find details of this assay. Since I was interested in that, I wanted to see if I can adapt this assay from the microscopy again to flow cytometry, if I can detect microRNAs with flow cytometry. Now, we're interested in T cells, and I was really inspired by this really nice paper on JXMed by Mark Ansel's group, where what they did is they took T cells, they stimulated them with CD3, CD28, and they performed microRNA profiles of the T cells. And what they realized is that after T cell stimulation, the majority of microRNAs, they actually get downregulated. Now, the primary role of microRNAs is to suppress mRNA. So when you stimulate your T cell, you will want some mRNAs to be upregulated in order to have, T cell function. So the majority of microRNAs were actually downregulated. However, there was a group of microRNAs that was upregulated, and the best hit on the list was microRNA 155. And what was later proven throughout the studies that microRNA 155 basically targets inhibitors of the TCR signaling. So microRNA suppresses the inhibitors and basically allows the TCR signaling to go through. So when you have high levels of microRNA 155, you have better signaling through the T cells. And this was of great interest to us because of the studies that we're doing in T cell immunology. And we embarked upon trying to prove whether this assay works with detection of miR155. So what you can see here, we chose microRNA 21 as a control because it was shown that it's not upregulated after T cell stimulation, at least not at the time points that we were investigating. And we investigate overnight stimulation here with PMA and oomycin. So you have here T cells with CD4 T cells with no stimulations. Cells will not express CD61 as much, and there is basically no detection of microRNAs. When we stimulate with PMA and oomycin, you see upregulation of of CD69. But if you see here on the histogram, we don't see really an upregulation in the expression of miR21, microRNA 21. Now, the story changes when we go and look at microRNA 155, where again, with no stimulation, we don't detect much. But when we stimulate with PMA and oomycin, we now see a nice upregulation of microRNA that really correlates with upregulation of CD69. And it's even more obvious when you see the histogram, where basically the whole population shifted towards miR155 expression. So this was really interesting and really exciting when we did the experiments. We performed several set of experiments on 4 subjects. We've now done even more subjects. And you can see that after PMA and oomycin stimulation, we can detect upregulation of miR155 in both CD4 and CD8 T cells. And we've done now that with other stimuli as well, not just PMA and oomycin. You can see again this data on the new paper that we got recently out. So I think the advantages of this assay is really shown in this 3D picture, where you can really do simultaneous detection of microRNAs, mRNAs, and protein at the single cell level. And it's very high throughput. People that have used flow cytometry, they know how quickly it is to acquire millions of cells within a few minutes. And basically, again, just to characterize it, we were able to detect miR155 expression with IL-2, mRNA expression, and in combination with protein. And you can do that really at the single cell level. And because you can use now more antibodies, you can go and characterize these cell subsets and see what sort of other markers they express. Now, people ask how sensitive this assay is, and this is a kind of complicated assay answer. The reality is that depending on the probe design, depending on how big your transcript is, so the bigger the transcript, the better, the more probes you can actually target. What the company did, Affymetrix did this experiment. It's not an experiment that we did in our lab. You can actually see that you can detect down to 10 copies with confocal microscopy, and then we can actually see nice separation between the irrelevant probe the gene of interest, which in this instance HMBS. So they can confidently say that in this type of with this gene of interest and with this type of probe, it's Alexa's 647, they can confidently go down to 10 mRNA molecules. The reality is that scientists should really go and check their own the sensitivity of the assay for their interest for the mRNA of their own interest. Now, just to finish here, I just wanted to say that using this assay, we're able to use microRNA to detect microRNA and mRNA detection at the single cell level with flow cytometry. I have to say now that the detection of microRNAs with flow cytometry is not currently supported by Affymetrix. These are experiments that we did in the lab. They're working towards that, but it's not currently supported available for customers. We showed that we can have detection of mRNA in rare populations such as HIV and CIV specific T cells. We showed that this assay is compatible with antibody staining and viability diet, something that is really important when looking at human samples or samples from tissues. We showed that detection, we were able to detect targets for which no antibody currently exists or there is poor staining. This assay is really simple. The main idea is similar to intracellular staining, so it's ideal for immunologists. It's very standardized. The probes are available from the company, so you don't have to design anything. And basically, the protocols are very standardized and very well characterized. And it's basically ideal also for animal models with poor antibody database. In our group, we have perform experiments with non-human primate samples, so we know that if we can design probes for non-human primates, they work. The probes also work with mice and other specificities. And finally, I just want to mention, as I said, that the majority of the experiments that I showed you here is with the quantity gene. It's the first generation of buffers and kit that Affymetrix produced. Now they have even more advanced version that came out. It's the PrimeFlow. We are working with this assay in the lab. I couldn't show you some of the data because they are unpublished. This assay is methanol-free, so you can use a greater variety of fluorochromes. It is now compatible with intracellular antibody staining, so you can look for transcription factors or cytokines. And it basically has a complete buffer system and is a more stable system. It's an advanced version of this assay, and we're really excited to be able to work with it. So at the end, I just want to highlight and acknowledge all the people that work in this, people from the Reagan Institute, and especially Daniel Kavanagh that we collaborated at the beginning, and Bruce Walker, who is the director of the Reagan Institute. I want to have special thanks to Daniel Kaufman. He's the last author of the Nature Communication paper. He was my mentor while I was doing my postdoc, and he basically provided the environment and the expertise in immunology to perform these studies. Scott helped with the ImageStream technologies he's working at MGH, and a lot of thanks to Affymetrix and eBioscience, especially to Kwan, who was a person who we started working when he was in Panomics, but special thanks to Steve. He has been a very, very, very important collaborator, and he was standby next to him basically throughout all these experiments, and also the funding sources and the different agencies that work. So I would like to stop here, and thank you very much for listening. And I will pass now the control to Judy.
Thank you for that informative presentation. Before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. And our first question is from Nasir Umar Safet, DNA Labs. Could this technology be applied to viral research in such a way that it could complement RNA viral loads?
Well, I don't know if we are there, but we can definitely detect viral RNA. Down the line, if the assay is optimized accurately, it could actually work towards that, especially for viruses that are very difficult to detect. It is something that is very useful, for example, when we look at tissues like lymph nodes, and you want to look at virus concentration, viral loads in either lymph nodes or tissues that you cannot do a regular assay. So this assay potentially down the line has great potential for this. So it's a great question, and we haven't worked on that yet, but this is something that we're really interested in down the line. That's actually how we started with Daniel Kavanagh, being able to detect viruses in different cell populations. So down the line, potentially, yes, we still need some optimization, but yes, it could be one of the future applications.
Okay, then from Ian Dinnick at NCLE University, after labeling, how quickly must you analyze on the flow cytometer? And in combination with that, Giorgio Mangino from Sapienza University of Rome asks, which fluorochrome are type 1 and type 6 fluorochromes?
So once you finish the whole essay, you basically, Affymetrix in bioscience, they have this storage buffer that you can use. We usually go on the flow cytometer straight away, but we have done experiments where we kept the cells 1 or 2 or even 3 days in the fridge and went afterwards to detect, and detection was pretty good. I can't say that I have done extensive qualitative analysis of the difference in how much signal you may be losing. We detected nicely. I think perhaps the company will have a better answer, and they have a very nice system where if you send them an email, they will definitely reply quick. So I think it's worth asking them. And now when it comes to the second question, type 1 is Alexa 488, type 4 is Alexa 647, and type 6 is an Alexa 750. So your flow cytometer need to be able to detect those. Most cytometers are able to detect at least 488 and 647. So thank you.
Dimas Abdirama of Charité Universitat Medizin Berlin asks, could this technique be used in combination with intracellular staining to detect transcription factors? Because methanol-based buffer could also destroy the protein structure of transcription factors.
Yes, that is an excellent question. And so, as I mentioned in the end, the first version of the kit, of the buffer set, of the kit that we were using, could not do that. But as I mentioned, Affymetrix came up now with a new generation of buffers. They're really good, and you can now, it's not methanol based, so you can now actually detect transcription factors inside the cells with antibodies, and it gives you great possibilities to do a lot of functional work. So PrimeRNA is now the best option to go if you want to detect transcription factors.
Wei Zhang from Medical University of South Carolina asks, intracellular cytokine analysis by flow cytometry is well set up. What is the advantage of RNA probe?
Well, as I try to highlight throughout my talk, yes, intracellular cytokine staining with antibody-based is very well set up. However, first of all, you block, as I mentioned, the cytokines inside the cell. So if you want to do functional assays where you want to see the impact of these cytokines, you cannot actually do that. The other thing is that because of antibody availability is heavily based, for example, you're looking at T cells, it is heavily based towards Th1 cytokines. For human samples, for example, when you want to look at HIV-specific responses, you can detect interferon gamma, TNF-alpha, IL-2, say MIP-1 beta. There are no good Th2 cytokine antibodies for Th2 detection, or at least the system doesn't work very nicely, especially for rare population. Then you have other cytokines that you may be able to want to detect, like IL-23 or some transcripts of IL-17 or other antibodies or other cytokines for which you just don't have any antibody available. And also, as I mentioned before, there are different transcripts of cytokines that may actually play a role in the pathologic condition. So with antibody, you cannot do that. You can just antibody-based techniques. You can just measure the protein that is based on the best antibody that you have. But now you can design probes for these different transcripts, and if expression of this transcript is high enough, you can actually distinguish between different transcripts of your cytokine. So I can see a lot of advantages apart from just being more sensitive for a few cytokines or doing in vitro assays. You can actually now detect different transcripts. So there are a lot of advantages of doing staining with this assay for cytokines. Obviously, the advantage is not just staying with cytokines. You can go and look other stuff that there's no antibodies, such as, I don't know, transcription factors or other molecules that are inside the cell that you just have no antibodies to stain with.
Okay, Wafa Comrie from Imperial College asks, how about the time point for protein ICS and equivalent RNA detection, especially when it is an induced expression? One would expect the RNA detection to be at earlier time points, and therefore it seems that the RNA detection has to be in a different prep of cells at a different time point, and therefore would you advise a time course experiment first?
This is a very good question, and when it comes to detecting different mRNA transcripts, especially cytokines, we need to, as I said, readjust the way of thinking, not just think in terms of ICS, but think in terms of kinetics of different proteins based on different cell types and different transcription, the different genes that we want to look at. So I definitely advise to do kinetics before you embark on going and do, I mean, you can use this assay to do the kinetics, but the easiest way to do the kinetic with qPCR on your bulk cells, it's faster, and you can do it. And once you see the best optimal time point for the gene of interest, then you can go and do further analysis at the single cell level with this assay. We have done that several times. We had a blood paper in 2011 where I'm first author, where we did kinetics after HIV GAG stimulation, and we were able to see the kinetics. And yes, mRNA does come first after the protein, but as I said before, in our hands, for example, IL-2 mRNA came first, and after like a few hours, that's when interferon gamma came up, and then the expression of IL-2 RNA went down. So you really need to optimize the time point that you want to look if you want to look at two different transcripts at the same time. So this is a very good question, and I definitely would advise to perform kinetics before you want to do functional assays with cytokines after stimulation.
Angelica Quapio from MUW asks, detection of gene-specific mRNA can be done for transcription factors such as well?
Yes, we haven't done it ourselves in the lab. This is something that we are really interested in, but I know other groups and other collaborators have done that. Yes, the other collaborators have done that. The studies are not published yet, so I cannot mention more, but this is definitely doable. Basically, any transcript of interest, if the expression is at decent levels, then you know, as I said, down to 10 copies, you can easily detect house-skipping genes. So if any gene of interest, you can design probes. The company actually designs the probes. You don't even have to do it yourself. You can just send them an email, and then you have the probes designed for you. You can actually detect transcription factors and anything that is of interest.
Inez Fritas from UCD CRID asks, can you explain a little better the co-localization studies? What are you measuring exactly?
So for the co-localization, so for the co-localization studies that we did here, these were just proof of principle experiments. So we were not really interested in co-localization, but basically what it measures here is the fluorescence of the RNA based the fluorescence of the protein. And you can, you know, I'm not an expert in that, but you can see here Amy Baxter and Scott Monterguy from MGH Scott did the ImageStream for us, and Amy from Canada, from University of Montreal, did the analysis. But basically, what we did here, you can see we use a similarity score or the bright detail similarity score, and we have detailed explanation of what we try to do in the Nature Communication paper. But what you can see here is that, for example, CD3 and CD4 co-localizes, so the two proteins are found at least in the same vicinity in the cells, while mRNA for interferon gamma and mRNA and protein for interferon gamma are not found in the same compartment. As I mentioned before, the way we perform this assay is with a capture of interferon gamma protein outside of the cells when it is released, while the mRNA is actually inside the cell. So there's no way in this experiment that the interferon gamma protein and the interferon gamma mRNA will be co-localized. However, there are other possibilities, depending on what sort of experiment you want to do, that people can use this ImageStream technology, or they can even go back to confocal microscopy, and they can see, for example, if an mRNA of interest can bind to the protein of interest, whether it's transcription factor or whatever it is that they want to do, or if a protein can bind to the mRNA, if it's a protein carrier of mRNA, or different styles that people can do. All we wanted to show here is a proof of principle that you can use this technique to now co-visualize the different mRNA or protein.
We have time for just one more question right now, and it will be from Angelica Quapio, again, MUW. Which flow cytometer did you use, and how many lasers?
This is a good question. So, as I said before, our group has great expertise in flow cytometry, and at the Reagan Institute we have a very good facility of flow cytometers. But we've basically used every cytometer we have. So we have a 5-laser for Tessa that we were able to detect up to 13 colors. We've used a 3-laser, again LSR. We've even used the FACS scan that we have upstairs. We've used the 4-laser. So basically the regular flow cytometers that people regularly have in the labs. And we've used the ImageStream as well, which is a kind of flow cytometer. So in different machines. So it doesn't need a specialized instrument to detect these.
Unfortunately, we are out of time. I want to thank the audience for their interesting questions and participation in today's event. We will answer questions via email that we did not have time to respond to today. I'd also like to thank our sponsor, eBioscience, for making today's educational webcast possible. Today's webcast will be available for on-demand viewing through June 2015. You'll receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time. Goodbye.